Hello. So I can't play any games unless I have people actually typing the code in to play the games. So welcome. We're going to start out with a game on Heredity. So if you will type in that game, go to joinmyquiz.com, and then you type in that code. You'll be able to log into the game, and then it will actually show up on your device. Like I'll have the main screen on so we can kind of see what the questions and the answers are, so we can kind of go over them together. But the actual game will show up on your side, where it'll ask you the questions on your side. You'll be able to answer on your side. That's why I like Quizizz, because I'm able to send the questions straight to your devices, which I haven't found to be the case with some of the other programs, like Kahoot. Like I love Kahoot, but there is a character limit with Kahoot, which makes me upset. So I just play the Quizizz games for my games that I've built everything with. So again, I can't actually start this game unless I have someone that's logged in it. I do see that someone has finally logged in. Yes, thank you so much. So just for give a couple moments. I just started the live, so I'm gonna say give like another minute or so, that if anyone else wants to join in on the game, they can still type in that code. So joinmyquiz.com. The code is 930056. And also, if anyone will do me a favor and type in that code into the question box, then I'll be able to pin the question so that if anyone like comes in later, they'll still be able to get the code and they can join in on the game on their own devices. So that'd be awesome if someone would just type that code into the question box. Again, that the code is 930056. If you don't want to type in the code, that's perfectly fine. I understand. You can play on your device, just kind of mentally thinking of what you think the answer is. There's a timer on each of them, and I kind of will wait until everyone that's playing the game online completes the question before I move on. So that will allow you to, oh yay, someone asked the question. You are the best. Thank you, Online Sucks. So we got that up in there. I'm going to say just a couple moments that if anyone else wants to join in on our game, they'll be able to. Right now, there is only one person in the game. We're going to start with Heredity. I've got the code pinned as well as I've got the code on the screen, 930056. And I'm going to start that game in just a few moments so that those of you that want to play can play. If I can zoom out a little bit. My setup in my closet is a little fun. I don't know if you can see like the corner of it, but I'm literally leaning these on like high-heeled shoes because that's just the fun way to do it. Okay, so let's zoom back out a little bit so we can press the start button. And I can show the game questions here as well so that those that are playing at home that aren't in on the game can still see the questions. Get a little better view for you. Monohybrid Cross, complete dominance. What's the phenotypic ratio? And you'll see questions like this on the exam where they'll tell you some information. Instead of doing all the math of a Punnett square, if you just know some of these ratios off the top of your head, it's just a little bit easier. So in monohybrid cross and with complete dominance, you're going to see a 3-to-1. Complete dominance refers to that the homozygous dominant and the heterozygous will both show the dominant trait, because just having one dominant allele will allow you to have that dominant trait. So 3-to-1 is our correct answer. Great job, y'all. So our next question. So if we're looking at incomplete dominance, what's our phenotypic ratio? And if y'all have questions as we're playing, you're more than welcome to type them into the chat, and I can answer them sideways as we go. My phone is a little sideways, so kind of makes life a little interesting. Okay, so when we're looking at incomplete dominance, that has to do with that the two alleles are not more dominant than the other. So you'll see a blending of the middle. So you'll have homozygous dominant being one phenotype. You'll have the heterozygous being a second phenotype. And then you'll have homozygous recessive being a third phenotype. So we see a 1 to 2 to 1 ratio because that's the homozygous dominant. The 2 are the heterozygous. And the 1 is a homozygous recessive. So we should see a 1 to 2 to 1. Great job, y'all. So if we're looking at a dihybrid cross, what is the probability of complete dominance? Well, what's the ratio with complete dominance? And so these are a little harder to do if you were to try to do that Punnett square out real fast. So knowing these ratios should make it a little bit easier. And the reason why I say to know the ratios, at least these main ratios, is because you don't want to waste your time on the exam going through a dihybrid cross. If you know these ratios, it makes life a lot faster and a lot easier. So when we're looking at a dihybrid cross, that means that we have two individuals that are heterozygous for two traits. So that would be you could use the math if you wanted to, whereas a 3/4 times 3/4 gives you a 9/16. 3/4 times 1/4 gives you a 3/16. 1/4 times 3/4 gives you a 3/16, and 1/4 times 1/4 gives you 1/16. So that's a 9 to 3 to 3 to 1 ratio since we're looking at complete dominance. Ooh, I spooked you on that one. The 12 to 4 would have shown if there was independent assortment. Or sorry, there's no independent assortment. They would have been linked together, which would have been a problem. So how many would you expect to be homozygous dominant and a cross between a homozygous dominant and a homozygous recessive? So considering the first parent is homozygous dominant and the second parent is homozygous recessive, we know that 100% of those offspring are going to be heterozygous because the only thing the homozygous dominant can pass on is a dominant trait. And the only thing a homozygous recessive can pass on is a recessive trait. So we would expect there to be 0 of the offspring that are going to be homozygous dominant because of the fact that they are unable to get two dominant alleles because both parents didn't have that dominant allele. So 200 offspring, how many would you expect to be homozygous recessive in a cross between heterozygous and homozygous recessive? And yes, I give a very short amount of time for these. Just because to keep life going a little bit faster. Plus, you should be able to do that math relatively quickly. Okay, so if we're looking at a homozygous, a recessive, and a heterozygous, there's a 1/2 chance that heterozygous is going to pass on that recessive trait, and then a 100% chance the homozygous recessive will pass on that trait. So 50% of the offspring are going to be homozygous recessive. So if we're doing 50% of 200, that tells you that there should be 100 offspring that will be homozygous recessive. So cross between white and red was probably a pink. And I'm going to assume that this is incomplete dominant, but I don't remember because I didn't set it up that I didn't write it in the question, so I don't know what I was thinking when I wrote this. It could go either way. I really need to fix this question and add that in there. Hey, so I think what I was looking for here was the pink offspring being incomplete dominant. And so if that's the case, then the answer is going to be 100%. If I was going for that the pink offspring would be heterozygous and not actually one of the things, I don't know what I was going. Where was I doing on that one? Oh, that's incomplete dominance. It should have been 50%. Sorry, y'all. So if we have the white and the red, I remember we said there was a 1 to 2 to 1 ratio for the incomplete dominance, so 50% is our answer. I'm sorry, I was not thinking clearly when I said that. Sorry, y'all. Actually, no one's playing anyways. I'm losing followers. Oh no. Cross between white terminal and red axial. Probably a very red axial. So here we're looking at, I'm assuming that we're looking at complete dominance. So red is going to be completely dominant over white. So 3/4 would be red. And then 3/4 would be axial. So the answer should be 9/16. There we go. Yeah, they'll specify. A lot of these games were made in the middle of the night during virtual learning last year. So there's some like little things where I didn't like put extra words in there. Sometimes I need to go back to these games and fix some of those wordings. But they will specify on the exam which one it will be. So if we're looking at a cross between a type blood and a B type blood, probably of a O type blood. So we're going to assume that that A type and B type is going to be heterozygous. So they are IA little I and IB little I. And so there is a 1/2 chance of each of them passing on that recessive allele. So 1/2 times 1/2 gives you 1/4. You could do that with the Punnett square, but that's the quick math of how you would come up with that answer. The join code is 930055. It should be pinned in the room. 930055 is pinned at the bottom of the page to help you be able to join the game. I wonder what these little symbols by y'all's names are meaning. I tried to figure it out the other night, but couldn't figure it out. It's like a little 3 next to Heather's name. I don't understand it. So cross between 2 roan cows was probably of a white cow. So the roan cows are going to be heterozygous. So if we're looking at this monohybrid cross, and we're looking for probability of white, that would be a 1/4 chance because we have 2 heterozygous. And then so there's 1/4 chance of homozygous recessive when 2 heterozygous cross together. Oh, is it 6? I'm sorry. The code went away and then I repinned a new code because the other one went away. Sorry. Let me get rid of that thing then. That's the wrong code. There you go. Okay, I didn't have a chance to check it because I was trying to fix the code. So incomplete dominance involves heterozygous with an intermediate between the 2 traits. So this one should be your right answer because it's incomplete dominance. Codominance is like what we saw with blood type, where you can have the A allele and the B allele at the same time, which shows you AB blood, as well as the roan cow is an example of codominance because you see both it's a red and a white at the same time. And so since you can see both of the traits independently of each other, that's considered codominance. Versus when we think about the pink flower, that was incomplete dominance because it was a blending of the red and the white together. That was the last question and the quiz. Okay. So that's the end of that game. So I have a couple other games that we could play. Go ahead and in the chat, you can tell me which one you want to play. Here's our options. We can either do biotechnology, we could do DNA replication, transcription, translation, or operons. So again, the different options we can play for our next game is biotechnology, DNA replication, transcription, translation, or operons. So go ahead and vote in the chat for me. Tell me which one you want to play next. And I'll get that game, started and we can play that game next. And we'll just keep playing the games that I have until we we kind of get ourselves settled up. Okay, so it looks like that I've got operons in the chat. So we're going to press the continue button and get this pulled up for y'all. Let me zoom in that code so you can see a little bit better. So the code is, whoa. There we go. So 459709. 459709 is our code, and I cannot press the start button unless I get at least one person to join. So if someone will please type in that code, and we will do DNA replication after this game, because I see that Heather's asked for DNA replication after this. So I do see there's one person in the game. There's two people in the game, so I'll give you a moment to put your answer in there. I'm sorry, your code in there. And if someone will type that into the question box, so I'll be able to pin it. That code is 459709. Again, that code is 459709. So you can type that into the question box, so I'll be able to pin it in case someone comes in the middle of the game and wants to join the code. Yay! Thank you, Heather. Okay, so let's press the start button. Is it still zoomed in? I think it's still zoomed in. Sorry, trying to get it so that it's optimized to the best I can for the screen. There you go. Okay. Which one describes an operon that's inactive and becomes activated by a molecule binding to the repressor? Okay, so what we're looking at here is that the operon is originally starting out to be inactive, and then it becomes activated. So if something is becoming activated, but it was inactive originally, that's going to be an inducible operon. And I noticed before we got started, somebody said that they forgot what operons are. So real quick, operons are found only in prokaryotes. Set up with a couple different components. They have the promoter sequence, which is where RNA polymerase will bind. Within the promoter sequence is this area called the operator. The operator is where a repressor is going to bind. So the repressor will bind to the operator to repress or turn off the operon. We also have inducers. Inducers are going to basically allow for the repressor to become inactivated, so it pops off of that operator, so we can then, of course, turn it on. The big examples we have are a repressible operon would be a trip operon. So when tryptophan is present, we're going to stop making tryptophan. So the trip will bind to the repressor to turn off that operon. Versus in a Lac Operon, which is an inducible operon, the operon will not get turned on unless lactose is present. Because why am I going to make this up to make down lactose if lactose isn't there? Okay, so let's get back to the game. So which of the following describes an operon that is active and becomes inactive by a molecule binding to the repressor? You're welcome. I still don't know what these symbols are. Do y'all know why there's symbols next to y'all's names? There's like a little number 1 by Tori's name. No. Okay. So if the operon is on or activated and it becomes inactivated, that's going to be considered a repressible operon because I'm turning it off. I'm repressing this operon. So that would be your repressible operon. Next. So the Lac Operon is an example of inducible operon, which describes the process that occurs when lactose is present. I know they're kind of a lot to read. Sorry, y'all. That's why it's kind of good that it's on your device instead of my device. Those that are playing at home that are just using the screen, they might have a little trouble, but otherwise, you should be okay. Okay, so with our Lac Operon, the lactose is going to bind to a repressor, and by binding to the repressor, it inactivates it, so the repressor stops binding to the promoter. So that should be inactivates repressor, releases from the operon to turn the translation on. So it should have been that third answer choice. There we go. Hey, and you got it right. Great, go. Trip operon is an example of a repressible operon, which the following describes the process that occurs when trip is present. So in the trip operon, what we're looking at is a repressible operon. So it starts turned on, and it gets turned off by the tryptophan being present. So tryptophan is going to bind to the repressor. When it binds to the repressor, it's going to activate that repressor. So the repressor will then bind to the operator, which will inhibit RNA polymerase from binding, so it turns that operon off, so you don't translate. Great job. In the trip operon, tryptophan binds to the repressor, which following term is used to describe this molecule. So the molecule that's binding to the repressor. Okay, so since we're looking at the fact that it is a repressor and is turning off the repressible operon, we're going to call this a co-repressor because it's going to help the repressor to turn off and repress this operon. So we're looking at co-repressor. So here, I'm going to look at the Lac Operon. Allolactose binds to the repressor, which the following term is used to describe this molecule. Okay, so since the original Lac Operon is an inducible operon, allolactose is going to bind to the repressor, which is going to cause your repressor to become inactive. So it's going to induce or turn on your operon. So we call that an inducer. Enhancers are going to be found in our eukaryotes. So the enhancer is going to aid in the binding of your RNA polymerase to the promoter once the DNA does its wraparound. So like there's different binding proteins that will bind to the DNA that kind of causes it to fold over. And it creates like, I almost think of it like a baseball glove. It makes like a nice secure fit for RNA polymerase to bind to that promoter. So it helps out. Which following describes a pathway that the inducible operon controls. And this gives us a moment to kind of think about what's the difference between anabolic and catabolic. So anabolic means that it is going to be building up something. So we're starting with small molecules and we're using energy to make a big macromolecule or a big molecule. Catabolic has to do with that we have some big complex molecule and we're breaking it up into small molecules. So since we're looking at an inducible operon, it's making the materials to break down that lactose. So we're looking at catabolic. I think y'all were split in the middle. That's good. So the following describes pathway for a repressible operon. So in this case, when we're looking at the repressible operon, it's like our trip operon, where we're making the tryptophan. And if tryptophan is already there, we don't need to waste that energy to build it. So that would be an anabolic pathway because we're building something up from something smaller. Which of following describes the function of the operator in the operon? Okay, so since we're looking at the operator's job is to, it's in the middle of our promoter sequence, and that's where the repressor is going to bind to turn on and off the operon. The promoter is the site of RNA polymerase binding. That makes no sense because the promoter is a region of the operon, and then the inducer binds to the repressor. So it has to be that first one. Last question in our group. Which following describes the function of the promoter? Oops, I gave that away on the last question. Okay, so since we're looking at what's the function of the promoter is the site where RNA polymerase is going to bind for transcription. We'd see this in both prokaryotes and eukaryotes, but the whole operon is only going to be found in our prokaryotes. So the answer is site of RNA polymerase binding. The operator is already a region within the promoter, and then the inducer is what's going to bind to the repressor, so not the answer. And that's the last question for that game. Okay, so that we said that we were going to do DNA replication next. So let's make sure I have all the settings. Timers. Okay, we're good. Okay, let's zoom this up for y'all so you can feel better. Okay, so there's the code. And we've already unpinned the last one. So our code is 058210. So that can allow you to join the game. Remember, I can't press the go button on the game unless someone is in it. I wish I could. That's okay. Sooner or later, someone will join the game, right? 058210. Is this helpful for y'all, by the way? Aw, thank you. I don't know your teacher personally, but I hope that the resources have been helpful for you. Again, that game is 058210. And if someone will type it into the question box so I can pin it, that'd be great. That code is 058210. So if you can write that in the question box, that will help me so I can pin it. So 058210. That's good. Okay. Yeah, and we're still a good 6.5 weeks away from the AP exam. It's just a matter of me trying to get all the review in and not overwhelming it myself and y'all. So I kind of spread it out over a very long period of time. That's why I do like daily review on my Instagram. Okay, so no one's gonna type it into the question box for me, please. 058210. I really appreciate it. Okay. So I guess if someone asks me the code while we're playing, I'll be able to do it later. Oh wait, I gotta super zoomed in it. Okay, which of the following enzymes are responsible for synthesizing the new DNA strand? Which of the enzymes? Okay, so the enzyme that's responsible for synthesizing new DNA, enzymes always tell us what they do, right? So DNA polymerase is going to make a new DNA polymer. Helicase is going to break the helix, so it breaks those hydrogen bonds that are between the nitrogenous bases. Primase makes an RNA primer, and DNA ligase kind of ligates or like puts things together. So the answer for this one about which one is making new strands is DNA polymerase. Nice job, y'all. So what's the difference between a leading strand and a lagging strand? Okay, so the right answer here is that DNA is synthesized continuously on the leading strand and discontinuously on the lagging strand. A lot of students kind of get confused over here thinking that the directionality is different. DNA polymerase can only add to an open 3' end. So DNA synthesis is always going to take place from a 5' to a 3' directionality. The 5' is where our phosphate group is, and the 3' is where our hydroxyl group is. So it's always going to be made 5' to 3'. The difference between the leading strand and the lagging strand is because DNA is anti-parallel. The leading strand is the strand that's going to be replicated as it moves into the replication fork. So as helicase is unzipping or breaking apart those hydrogen bonds to split apart our DNA double helix, the leading strand just moves continuously toward that splitting apart. While the lagging strand is going to move to where the open is, and it's going to work backwards. Because DNA is anti-parallel, one of them has to move backwards or one of them moves forwards. But they're still 5' to 3'. We all had that. Great job. I went through that explanation for nothing. Sorry, y'all. The following enzymes is responsible for joining the Okazaki fragments. So Okazaki fragments are the short fragments that we're going to make on our lagging strand. Because the fact that it's going to be made discontinuously, we have all these little fragments, and so DNA ligase is going to seal those together. As we said before, tells you what it does. DNA ligase ligates or puts things together, creates that bond, that phosphodiester linkage. Which the following explains the base pairing rules. And this kind of trips students up sometimes too. Okay, so it's very simple to remember which ones go together. So A pairs with T and C pairs with G. So that's not usually where students get confused. There's different ways you can remember it. I teach in Conyers, Georgia. So I remember like at Conyers, Georgia. I graduated from Georgia College, so I go at Georgia College. So just find a way to kind of help yourself to remember the A, T, A, and then the C and the G will go together. The trouble that students have is remembering the number of bonds, and this is directly out of the CED to know the number of bonds. So A and T will have 2 bonds, and C and G will have 3 bonds. Way to remember that is C is your 3rd letter in the alphabet, and you need 2 lines to make a T. I really don't have any other way to remember that. That's just kind of the way that I've made up to help students remember it. Yeah, and when y'all already knew that, why do I keep explaining things? Y'all are on the ball. Sorry, y'all. Following enzymes responsible for breaking those hydrogen bonds between the nucleotides. Okay, so the one that's going to break those hydrogen bonds is going to be helicase. It's going to break apart the helix. Remember, enzymes tell you what they do. So helicase is going to break that double helix. It's going to break up those hydrogen bonds to unzip or split apart those genes. So which following explains why DNA polymerase requires a primase for synthesis of a new strand. Yeah. Okay, so in this case, primase is going to make that RNA primer. So the reason why we need to have an RNA primer before we can go through DNA synthesis is because of the fact that DNA polymerase requires an open 3' end. It can't build off of nothing. So have you ever heard of like the way that crystals are formed? In order to make a crystal, you have to put like one piece of sand in there in order for that crystal to form. It just needs something to build off of. And so it needs that open 3' end. It wouldn't be 5' because DNA is made 5' to 3', so that 3' end is the side that's going to add to it. And yay, we got a combination there. Hey, somebody figured out the code. Good job, y'all. Didn't even have it posted. Okay. Which one describes the 5' end? So what is at the 5' end? Okay, so at our 5' end is where we are going to have the phosphate group. So if you think about the structure in your deoxyribose or your ribose, and you were to count the carbons around, the first carbon is where you're going to find the nitrogenous base. Your second carbon is where you see the difference between DNA versus RNA, because ribose will have an oxygen there, while deoxyribose won't have an oxygen at that second carbon. The third carbon just has the hydroxyl group that's normally in a sugar. There's nothing major on the fourth carbon. The fifth carbon is where we'll see that phosphate group attached. So whenever they're talking about like the 5', 3', they're just numbering the carbons. It's not really important for you to be able to number those carbons, but you do need to understand the 5' end has a phosphate and the 3' end has a hydroxyl group. So difference between prokaryotes versus eukaryotes in terms of replication. Okay, so prokaryotes are going to have circular DNA versus eukaryotes have linear DNA. And so because the prokaryote is a circle, if you have one side, one origin replication, you can start at that origin and move the whole way around the circle. So you'll see that they only have one origin replication versus eukaryotes are going to have multiple origins replication. Number one, because they have so many strands of DNA, and number two, because it is more efficient for them to have multiple origins to go through all of that DNA because they have significantly more DNA in a eukaryote than you do in a prokaryote. Which one describes why there's a leading and a lagging strand in DNA replication. And I mentioned this earlier if you were paying attention. So remember, the leading strand is the one that's moving continuously into the replication fork, and the lagging strand is the one that's moving discontinuously. So the reason why we have to have a leading and a lagging is because DNA is anti-parallel. So only one of those two initial strands, one of those two template strands, is going to be in the appropriate 3' to 5' orientation to be read. Otherwise, the other one has to move backwards in order to still be reading 3 to 5 to synthesize 5 to 3, because anti-parallel. Hey, you got it. Way to go. I think this is our last question, and I'll turn the camera around and talk about DNA replication for a second. Which following strands would be more stable at a higher temperature? And of course, this is like looking at a double strand, of course, so you can assume what the base pair is on that. So we mentioned earlier the number of hydrogen bonds between them. So hydrogen bonds are one of those like weak bonds that if you have a lot of them, it increases their stability and their structure and their how strong they are. So we mentioned that adenine and thymine only had 2 hydrogen bonds versus cytosine and guanine had 3 hydrogen bonds. So you're going to kind of look through these to figure out which one has the most cytosine and guanine bonding together. And so this one right here, we have all cytosines and guanines in it. So this one's going to have more hydrogen bonds. So this one would be our most stable at a higher temperature that would traditionally break those bonds. Great job, y'all. Okay, and that would be the last of that question. So somebody in the chat asked if I could go over replication for a moment. I'm sitting in my closet. I'm going to flip the camera and I'll turn on the camera. I'll flip my camera and I'll turn on my, where is my camera? How do I flip? How do I? Maybe I won't flip my camera. Enhance? Haha. Flip my camera. So you're going to see awkwardness because I'm like in my closet, but whatever. Okay, so hi. So with DNA replication, there's a couple things to know, right? So there is going to be the origin replication. So there's a starting point, right? So the first thing that's going to happen is you're going to have helicase. Helicase is going to unzip or break those hydrogen bonds that you'll see between the two strands, okay? DNA polymerase is primase is going to bind and it's going to start to read it. It reads it 3' to 5', so they can pair up 5' to 3', like, because it has to make 5 to 3. Okay. After the primase makes its little about 5 to 10 nucleotides long, you're then going to have DNA polymerase building off that 3' end and start building and moving in, right? So it's going to just base pair up everywhere where it sees an A, it puts a T, everywhere there's a C, it puts a G, and so on and so forth, and it just builds that new strand. And so you're going to have something called single-stranded binding protein. Single-stranded binding protein is going to bind to the unbound strand to stop them from coming back together. They're hydrogen bonds. They're just attractive bonds. So they come right back together. So we need single-stranded binding protein to bind to them. You're also going to see that we'll have the helicase as before. That's going to break those hydrogen bonds. And then you have top isomerase. Top isomerase is like downstream, and it's going to relieve that strain. So if you think about like shoes and you were to twist your shoelaces up and then you pull them apart, you'll see that it gets like super coiled at the end. So that's the same thing that would happen with DNA. If you just pull apart the two strands, we create a super coil upstream. So top isomerase is going to relieve that super coiling that's going to take place upstream to do that. And then, of course, there's a leading strand. As I said before, it's going to move continuously into the replication fork. So if this was my DNA, it kind of comes apart like this, right? Because here's my replication fork right here. So my leading strand is going to move directly into my replication fork. And as the strand continues to open up, it's going to keep on moving in toward that replication fork. Versus the lagging strand, if I open up a little bit, it comes backwards this amount, right? And then if I open up a little bit more, it then comes backwards this amount. And then you need the DNA ligase to seal those little pieces together and create that phosphodiester linkage. You would need the same DNA ligase in order to seal together the little fragments we'll make in eukaryotic DNA anyways with the leading strand, because of the fact that there's multiple origins replication, but you still will use that. Is there anything else I'm missing? I think I got it. Hopefully that was a good, what you needed to hear about the overall process. So I'm going to figure out how to flip my camera back. Oop, I want to send this to someone. Flip camera. Yes, I have like the craziest setup right now in my closet. Just for you all. Okay, let's turn the lights back off. Okay, so our next game that we can play, we have either we could do biotechnology, we can do transcription, we can do translation. I think those are your options. So biotech, transcription, translation. So can you type in the chat which one? The lagging strand is still going to go 5' to 3' in terms of making it. We're still going to synthesize 5 to 3. So I don't understand what you mean the directionality. Directionality is always going to be 5 to 3. You have to kind of look at the strands. I can't show you any of the secure questions, but there is a question that has been on an exam. I think it was maybe the 2014 exam, where you were given a DNA strand and you had to identify which one was correct. So you're looking for that 3' end. And making sure that you are reading a 3' to 5' and making it 5 to 3. And so that was what they were testing on that question. DNA polymerase forms those hydrogen bonds for the RNA. No. So DNA polymerase is going to form the phosphodiester linkage between the nucleotides. So each of the nucleotides will come in, the adenine nucleotide, the thymine nucleotide, the cytosine nucleotide, and the guanine nucleotide. As each of those nucleotides come in, it will create the bond between the long chain and that new strand, that new nucleotide that came in. And the RNA, yes. So RNA can be replaced by one of the versions of DNA polymerase. So there's a couple different types of DNA polymerase. You are not required to know the different types of DNA polymerase. You just have to be able to know that DNA polymerase is responsible for making that DNA polymer. But part of the proofreading that takes place with replication is that DNA polymerase can come in, take out wrong strands and put them back in, and also can replace the primase that's already in there, because you don't want a whole bunch of RNA in a strand. No one has told me which one we're doing. Do y'all want to do biotech next, or do you want to do transcription, or do you want to do translation? Please comment and tell me which one you would like to do, and then I will pick on that game. You're welcome. Which game do y'all want to do? Biotech, transcription, translation? Anybody? I have 3 games for us, and y'all have 10 minutes left, so we probably have the time for maybe 1 game, maybe 2. That is not a word. Translation. Okay, so let's go look at translation. We have 2 votes for translation. Okay, so let me zoom this so that y'all can be able to see it, because I know it gets a little small on your screens. So, ooh, sorry. Okay, so the code is 134001. I still don't know why some of you have little symbols by your name. That really doesn't make much sense. I wonder if it's because you follow me. What if the people who follow me have like a symbol, and the ones who don't follow me don't have a symbol? That might be a reason why there's a symbol or not. Okay, so code is 134001. If you want to type that code and join our game. I'm thinking this will probably be our last game, just because there's only about 10 minutes left. And I have to go take care of my children. Yay! Okay, so I see there's 1 person in. Does anybody else want to join the game? 134001. Going once. Going twice. Sold to me pressing the start button. So, which of the following is the site of translation? Trying to find the right code. There it is. That's what I need. Oh, you're welcome. Happy to help you all. We all need somebody in our corner. Okay, so the site of translation is going to be your ribosomes. That's where we see protein synthesis, and that's what translation is. Translation is just reading an mRNA strand and then synthesizing a polypeptide from it. And so protein synthesis takes place in the ribosomes. Nucleus is where we're going to find our DNA. That's also where we're going to have transcription take place, where you read that DNA and you synthesize that mRNA strand. The Golgi bodies are responsible for packaging and modifying the products of the rough ER. And the cytosol is just the jelly substance that everything just kind of sits in. So the answer for this would be ribosome. Which technically I see that logic that you put cytosol, but there are 2 different types of ribosomes. Some are free and some are bound. And so if you were talking about a free ribosome, yes, the ribosome is in the cytosol, but it's not actually the cytosol that will have that protein synthesis. It's the ribosome that's in the cytosol. So I do see that logic where you selected the answer, though. So which of the following is our start codon? And no, this is not going to be asked on the exam. This is just me helping you, because if you know your start codons and your stop codons, it's just kind of helping to increase your efficiency on the exam. You won't have to worry about searching for things on your codon chart. You're not going to have some of that worrisome that might take place. You're not required to know the actual codon, though. But for those that want to know, the start codon is going to be AUG. A way to remember that is that school traditionally starts in August. I know some of our students are Labor Day students, and they kind of start after Labor Day. But so for my students, which actually is also a lie, school starts in August. So AUG is the start of the year. So start codon. I tell my students also, because the world revolves around me and I was born in August. So yeah, no, it works like that too. The function of our P site in the large subunit. Okay, so there's 3 sites that you'll see in your ribosome. That's on the large subunit. So we have, of course, the small subunit will be at the bottom, and that's where it's going to hold your mRNA. And then you have a large subunit that's going to bind to the small subunit. And the large subunit has 3 faces. There's an A, a P, and an E. At the A site is where you're going to add the new amino acid. It's just going to be where the next codon is going to pair with the anticodon that's on your tRNA. The P site holds that growing polypeptide, so there's going to be a tRNA held there that has a really long polypeptide attached to it. And in the process of translation, we're going to break the bond that's holding that tRNA to the polypeptide to attach it to the new amino acid that's brought into that A site. And then the E site is where we're going to have the empty tRNA is going to exit. So after everything's shifted over, after we've gone through translocation, and the polypeptide is now on the tRNA that's in the P site, the T site, everything shifts over. And so now we have a tRNA that's empty, and so it's going to leave. So our P site is where we're going to hold that growing polypeptide. Next one. So which following describes the process of translation. So what are the steps we see in translation? The A site is going to hold the, is going to add the new amino acid. So it'll be where we see the open codon. That's basically for our next amino acid. And that tRNA is going to join in, go through the base pair matching. And then that's when we'll add the next amino acid in at that A site. Okay, so which is following describes the options. So we're going to see we have a codon recognition. So we're going to recognize our codon, the next one that's in the sequence. So that's when the tRNA comes in. The anticodon on the tRNA pairs up with the codon. If they don't base pair, of course, that tRNA keeps popping out until we get the correct anticodon to pair up with the codon. And as a reminder, it goes through base pairing rules. A pairs with U pairs with A, C pairs with G, and G pairs with C. Then we'll have our peptide bond formation. So that's when we're going to form the peptide bond between that polypeptide that's already there and our new amino acid. And then translocation is when everything shifts over. So what's in the A site moves to the P site. What's in the P site moves to the E site. Hey, a biology teacher came. Welcome. This is just us playing games. How'd you see? Nobody answered that question. Okay, so which follows the direction that the mRNA is read? So which way do you read the mRNA? So the mRNA is going to be read 5' to 3', which is opposite of the way that DNA is read. And so I like to try to help students remember that with replication and transcription, you're using the same language, which is our nucleic acids the whole time, versus when we look at translation, you're going between languages. Now you're going between the nucleic acid language to a polypeptide amino acid language. And so since we're looking at a different language, it's going to be read in the opposite direction or a different direction. So 5' to 3' is how you read your mRNA. So that's why we have that 5' cap on our mRNA, is to tell the ribosome where that 5' end is to start reading to be able to translate appropriately. There's a lot of different languages out there that read opposite the way that English is read. Hebrew is one of those examples. And if any of you all read anime, you know that you start at the back of your anime book and move forward. No, you don't read DNA 5' to 3'. You read DNA 3 to 5 and you synthesize DNA 5 to 3. They're always anti-parallel from each other, so it's kind of hard to kind of get your brain wrapped around it sometimes. But DNA is read 3' to 5' and made 5 to 3. RNA is read 5' to 3', and then the polypeptide is made N terminus to C terminus. So which of the two describe the orientation of the amino acids when they bond? This is also a question, there was not this question, but a question similar to this on a previous exam, where they're having you demonstrate that you understand where that peptide bond is being formed. So the peptide bond is always going to be formed between the carboxyl group of the first amino acid and the amine group of the second amino acid. So you're never going to see the two carboxyls bonding together, and you'll never see the two aminos bonding together. It's always going to be the carboxyl of one and the amino of the next to create that peptide bond between them. And because there's a directionality to our polypeptide that is N-terminus to C-terminus, it's going to be the carboxyl group of the first one binding with the amine group of the second one, which I believe is right here. There's very slight differences in the way that the answers were written, just to make sure that you understood. And I try to model some of the stuff that you'll see on the exam where they change like a couple words. Difference between bound and free ribosomes. Okay, so our bound ribosomes are going to be ribosomes that are attached to our roughy R. And they're going to synthesize the proteins that are needed for secretion, as well as proteins for the actual membrane. Versus our free ribosomes are going to be ribosomes that are found in the cytosol, and they make those cytosolic proteins or proteins that are used in the cytosol. All ribosomes start out being free ribosomes. They'll receive a signal that tells them to move to that roughy R. So the answer we're looking for here is that free ribosomes will synthesize proteins for the cytosol, while bound ribosomes synthesize proteins for the membrane or secretion. We have a couple more questions. I realize it's 5 o'clock and I've run over my time. I'm sorry, y'all. The following describes the function of the tRNA. So what does that tRNA do for us? Okay, so our tRNA is considered our transfer RNA. And so its job is to transfer the amino acid to the ribosome. So it's bringing the amino acid to the ribosome. mRNA is going to be your transcript for protein synthesis. rRNA is going to be the one that's making up the ribosome. And these are honestly me just making up things. There's a lot of that goes on. We're just make up stuff for these. Following describes the function of the mRNA. So our mRNA is considered our messenger RNA, and it's going to be the RNA that comes out of our nucleus. That is the sequence, the code, the recipe, the whatever you want to say, for making that polypeptide that will pair up with that tRNA. Nice job. And I believe this is our last question, and then I will be logging off, and y'all have a wonderful Sunday. Which of them describes the conversion factor between nucleic acids and proteins? Okay, so there are 3 nucleotides for every 1 amino acid. So if you remember our start codon, we said it was AUG. That would be 3 nucleotides, right? You have the adenine, the uracil, and the guanine. Those 3 nucleotides coded for methionine, which was the start codon, right? Was our start amino acid. So it's 3 nucleotides for 1 amino acid. So you should be able to do this conversion either way, where if they give you a gene length, you should be able to say the number of amino acids that would be coming from it, or the minimum number of amino acids that would come. Same thing as if they tell you the number of amino acids in a protein, you should be able to tell them the minimum nucleotides that you could be making from this. And that is the end. So if y'all don't have any other questions, I'm going to log off, and y'all are going to say, have a wonderful day. So is there anything you need from me before I log off and say, have a great day? Going once. Going twice. Sold to me, clicking the exit button. So I hope y'all have a wonderful day. Remember, AP Bio Penguins are dressed for success. Bye, y'all.